So this is a carboxyl group, and this is an amine group. And we know these are ionizable functional groups where they can act as acids. For example, this is what these functional groups look like in their protonated form, but they can also be in their deprotonated form. And then they can get protonated, so we know they're acids. So because they're acids, we know they have pKa values. And we know the lower the pKa of a functional group, the more acidic the functional group is. So therefore, we know carboxylic acid is more acidic than this amine. But what determines whether they're protonated or unprotonated? We know they can be protonated or unprotonated. So what determines whether they're protonated or unprotonated? Well, it's the pH of the solution. We know aqueous solutions can have pHs, which tell us about how acidic the solution is. And we know if we have a low pH, we have a very acidic solution with a lot of hydronium ions and a lot of hydrogen ions. So if we have a low pH, we have a very acidic solution. However, if we have a high pH of maybe 13, we have a very weakly acidic solution and a very basic solution with very little hydrogen ions. So again, the point is, pH is a characteristic of a solution. So this solution, this aqueous solution, has a pH. So when we talk about pHs, we're characterizing a solution. However, pKa is talking about functional groups. When we're talking about pKas, we're characterizing a functional group. These functional groups have pKas, which tell us about how acidic those functional group is, while a solution, this aqueous solution, has a pH, which tells us about how acidic the solution is and how many hydrogen ions there are in the solution. So it's the pH that determines whether these functional groups are protonated or unprotonated. And there's this magic rule. So again, we know they can be in their unprotonated form or protonated form. So what determines if they're protonated or unprotonated? Well, I recommend you memorize this rule. If you memorize this, you'll always know whether a functional group is protonated or deprotonated. So what you do is you compare the pH of the solution to the pK of the functional group, and that'll tell you if it's deprotonated or protonated. For example, let's do an example. Let's say we have a pH of 1. So we have a very acidic solution. What you do is you take the functional group, for example, let's focus on this carboxylic acid functional group, and you take the pKa of that functional group and you compare it to the pH of the solution that the functional group is in. And if the pKa of the functional group is higher than the pH of the solution, then that functional group will be in its protonated form. If the pKa of the functional group is higher than the pH of the solution that the functional group is in, then that functional group will be protonated. So the same thing with this functional group. It has a pKa of 9, which is higher than the pH of the solution it's in, so it will be in its protonated form. But let's do another example. Let's say we're in a pH of 7. We're in a neutral solution with a pH of 7. Well, we know this functional group has a pKa of 5, and we know now it's in a pH of 7, a solution with a pH of 7. So in this example, the pKa of the functional group is less than the pH of the solution. So when the pKa of the functional group is less than the pH of the solution, the functional group will be in its deprotonated form. But this functional group, which has a pKa of 9, the pKa of this functional group is still higher than the pH, so therefore it will be in its protonated form. That's why it's in its protonated form. So this simple trick will always tell you whether it's protonated or unprotonated. So let's do another example. Let's say we're at a pH of 11. Well, again, you take the pK of the functional group and compare it to the pH of the solution that's in. And again, if the pK is less than the pH of the solution, it's in its deprotonated form. So that's true for both of these functional groups, so therefore they're both in their deprotonated forms. And again, the way I like to specifically think about this is this. Essentially, if we're in a low pH, what does that mean? That means we're in a very acidic solution. And if we're in a very acidic solution, that means we have a lot of hydrogen ions. So if we're at a low pH, we're in an acidic solution with a lot of hydrogen ions. So if we have a lot of hydrogen ions, there's a lot of pressure from these hydrogen ions to deprotonate the functional groups, these functional groups. So therefore, when the pH is low, there's a lot of pressure from those hydrogen ions to protonate those functional groups. So therefore, the functional groups will be in their protonated forms. So that's how I like to remember. If the pH is low, there's a lot lower than the pKa. Therefore, there's a lot of hydrogen ions, so therefore the functional groups will be protonated. However, what happens when the pH is high? If the pH is high, it's low acidic. So therefore, there's very little hydrogen ions. So therefore, there's very little pressure from these hydrogen ions. So therefore, the functional groups will be unprotonated. So again, when the pH is higher than the pK of the functional group, it's in its deprotonated form. So that's the way I like to remember this rule. But the point is, whenever you have a situation, whenever you have the pK of a functional group, you just compare it to the pH of the solution it's in, and you follow this rule. And again, you can determine if that functional group is protonated or deprotonated. So now that we know this, let's talk about isoelectric focusing. So we can create this very interesting setup where we have this pH gradient. It's really interesting. So here we have a high hydrogen ion concentration with the low pH. Here we have a neutral pH. And here we have a very high pH with a very basic solution. So we create this hydrogen ion gradient where we have a lot of hydrogen ions here and then very little hydrogen ions down here. So we create this pH gradient. And not just that, we create these nodes where we have this positive charge here and this negative charge here. So now that we create this very interesting setup, what we can do is we can take, for example, a solution with these functional groups and dump it in this very interesting setup. So for example, let's say we had some kind of solution, this mixed solution with carboxylic acids and these amines. And let's dump it in this solution in the center of our interesting setup. What's going to happen? Well, we already explained at a pH of 7, this guy will be deprotonated and this guy will be protonated. We already explained at a pH of 7, so this guy will be deprotonated and this guy will be protonated. And we've explained that already. So now we dump these functional groups in the center place where we have a pH of 7. So again, this guy will be negatively charged and this guy will be positively charged. So what's going to happen? Well, this guy's negatively charged, it's going to attract towards his positive node. And if this guy's positively charged, it's going to attract towards his negative node. So now they're going to separate. So this guy's going to diffuse in this direction. This guy's going to diffuse in this direction. So they're going to start diffusing and start moving. So once they start moving, now they've separated. But again, at this pH, this guy's still negatively charged, so it's still going to attract. And at this pH of whatever, maybe 8 or 9 or whatever, it's still going to be positively charged, so it's still going to attract to this negative node. But eventually they're gonna keep moving, and eventually they're gonna move to a certain pH. For example, at a pH of 4, this guy will eventually get protonated. And again, we've explained that, and you can do these experiments and see at this pH with this pK of this functional group, we'll see it's protonated. And once it's protonated, now it's neutral, and once it's neutral, now it's not gonna move towards this positive charge anymore. And then also the same thing with this guy is once it gets to a pH of 10, it's also going to get deprotonated, and then this guy's going to become neutral. And once it becomes neutral, now it's not going to be attracted to this negative node anymore, this negative lead anymore. So now they're neutral, now they're going to stop moving. But notice what we've done. We've effectively isolated them. We've isolated them. Originally what we had was this solution, this mixed solution. However, if we dumped this mixed solution, we know they would start moving through this isoelectric focusing, and they would keep moving. Then eventually they would become neutralized and then they would stop moving, but we've effectively isolated them. Now we can just take the solution and now we have a pure carboxylic acid. So this process of separating these functional groups by creating this pH gradient is referred to as isoelectric focusing.